So far, it has been really difficult to identify the cause and effect relationships between hemodynamics and vascular tissue regeneration, and this is because it's challenging to control the individual mechanical loads. This bioreactor enables us to mechanistically investigate the individual and combined effects of shear stress and cyclic stretch on the regenerative potential of a broad variety of tissue-engineered vascular grafts. Demonstrating this procedure will be Suzanne Koch, a PhD candidate in our group, and Dr. Tamar Wissing, who is a postdoctoral researcher from our laboratory.
To mount the electrospun scaffold onto the silicone tubing, thread a 4-0 proline suture into one end of a piece of silicone tubing and out of the other. Make a small knot on both sides of the tube, leaving approximately 10 centimeters of wire on both knots, and make a third knot on top of the tube. After removing the suture needle, trim the edges of the silicone tubing into a triangular shape, and make a final knot at the end of the two 10-centimeter wires. Dip the electrospun scaffold in 30% ethanol, and place the scaffold over one end of free suture wire. Next, gently stretch both sides of the silicone tubing and the 10-centimeter suture knot, while a second researcher uses tweezers with a smooth inner tip to gently slide the electrospun scaffold over the tubing. Slowly release the stretch on the silicone tubing while simultaneously smoothing the electrospun scaffold with the tweezers, and dip the scaffold and silicone tubing in ultra-pure water 2 times. Construct the bottom compartment and make sure that the O-ring is properly placed. Place an adapter bushing in the upper part of the bottom compartment and place the pressure conduit with holes through the bottom compartment.